Welcome back, life science learners. So let's continue with today's installment of scientific methodology. Let's get straight into it and try and understand the process of scientific method. So let's look at what we've got on the slide. So it's important for us to know what is scientific method. So if we were to analyze this and unpack this concept, we would read as follows. The scientific method is defined as a method of research in which a problem is identified, relevant data is gathered, a hypothesis is formulated from this data, and the hypothesis is empirically tested. In simple terms, the scientific method is a way for scientists to study and learn things. So guys, if we were trying to establish what is the chance of us having heads and tails, we would need to come up with some kind of question. And that question is, what is the probability of having a head or a tail on a coin? And in order for us to be able to prove that it is 50%, we can guess that it's 50%, but we would need to test that. And so there is information that you know already that, a coin has two sides. There is a chance of it landing on a head or a tail every time you toss it. How do you present all of that in a research paper? Or how do you write a report that tossing a coin gives you a 50% chance of having a heads or a tail? So we would need to carry out a scientific method. And so we would need to write a question out, we would need to write a hypothesis out, and we would need to write an aim. So if I toss a coin, the question is, what is the chance of me having a head? And probably you're going to say, well, 50% or 1 in 2 chance. Can we test that? And so we would need to design a hypothesis. So my hypothesis is going to be, when I toss a coin, I have a 50% chance of having a head. Or when I toss a coin, I have a 100% chance of having a head. One of these two is going to be tested either positively correct, or we're going to get a negative result at the end. So we would need to review that hypothesis. So that would be the hypothesis. It has the independent variable, which is the chance of having a heads or tails, and the dependent variable would be the percentage chance. So I said either 50% chance of having a heads or 100% chance. So we're going to measure that. We also need to ensure that we record our results. So that would be the dependent variable. So I will toss this coin a few times and count the number of times I get a head and a tail. And I'll record that as my data. But before I do that, I need to write down an aim. So the aim would be the investigative question. When I toss a coin, what is the probability of me getting a head? So that's a simple question. And so I've got to carry out a method now that's going to generate data. And that's going to be the data that's going to talk to the hypothesis and answer the question around what is the aim of the investigation. So again, a simple concept, and you can apply that concept in most scenarios where you're trying to establish the scientific method to trying and solve a problem or question that you have no answer to. So let's go back to more processes around scientific methodology. So we've got to ask ourselves, what is a research question? And as I said, it's a question that can have answers that will generate a specific focus area. So a research question is an answerable inquiry into a specific concern or issue. It is the initial step in a research project. The initial step means after you have an idea of what you want to study. The research question is the first active step in the research project. So often at school, you're going to be given a research project. It could be on investigating the effect of pollution in your area. So you've got to understand, what is the focus of that research? So is it in your area? Is it in your street? Is it the littering of paper or plastic? So you've got to be very focused in that, so that your method becomes very defined and you're able to generate data that points specifically to your question. So it's important that you focus on a unique research question. And we often at school will ask our learners to come back to us and present their research question so that we have an understanding of their thinking and we can point you more specifically to a more pointed direction in your research. So it's often good to ask someone else, what do you think about my research question? Am I clear enough? Is this going to get me kind of information that I want to be able to answer my project or to be able to get information? So ask an intelligent research question, and that must be very pointed. Pointed in that it is clearly testing an aim. Okay. So here we get to the next bit. What is the aim of an investigation? So the aim identifies the purpose of the investigation. It is a straightforward expression of what the researcher is trying to find out from conducting an investigation. So with the example of, have you ever thought about why do plants grow upwards? We've got to write down a specific question that relates to the aim. So the aim would be to investigate why plants grow upwards. And that's very clear. It talks to the hypothesis, it talks to our research question as well. So the aim is essentially the intention of what you need to establish in your experiment. And that's very important, is that your aim should be very clear and relate to the intention of what you need to establish at the end of your investigation. Okay. What is the hypothesis of an investigation? So guys, often we get told that a hypothesis is a guess, it's a prediction. Essentially, a hypothesis is a statement around what you possibly think might happen, or what do you anticipate or predict that might happen. And the components of a hypothesis needs to include: one, the independent variable. It should include independent variable, and it needs to link to the research question. So, a hypothesis is a precise, testable statement of what the researcher predicts will be the outcome of the study. So, my prediction is that plants will grow upwards towards the light and increase in height. That's a testable statement. We would need to work around an aim and then a method to test that. So that's a statement that I predict that plants will grow upwards. Okay, so this usually involves the proposing of a possible relationship between the two variables, as I mentioned, the independent variable as well as the dependent variable. So what is the independent variable? This is the variable that the researcher changes. So this is what you have control over, and it's that variable that you're able to manipulate and change. So if I wanted to test why plants grow upwards, I have the ability to be able to control the factors that cause plants to grow upwards. Things like light, water, carbon dioxide. So those factors would be things that I have to manipulate. In this case, the dependent variable is what we're going to use to measure the results, but the independent variable is the variable that we are manipulating that has an impact or an effect on the other variables. So let's look at what that means. The dependent variable is what the researcher measures. And so if you think of measuring the height of a plant, measuring how quickly it grows, those are measurables that talk to results. And they tell us if there's an effect or there's an impact of the manipulating the independent variable. So the dependent variables often is what you measure that helps you to draw a conclusion or to be able to draw an inference from. So we got to know clearly that in our hypothesis we've got to include both the dependent variables and the independent variable. So what are the variables in a scientific investigation? And here we talk to specifically the types of variables. So we talk about the things that are changing in an experiment are called variables. So variables refer to variation. A variable is any factor or trait or condition that can exist in differing amounts or types. An experiment usually has three kinds of variables. As I mentioned, the independent, the dependent, and the control variables. And so we're going to unpack these as we get into the next slide. So the question is, what is the independent variable? Again, I'll explain this once we get through this slide. An independent variable is exactly what it sounds like. Again, a variable that stands alone. The word independent means standing alone. So it isn't changed by the other variables. So it is independent of the other variables and is not affected by the other variables. Okay, you are trying to measure this. So in terms of when you are carrying out an investigation, it's something that you can manipulate. So when I say manipulate, I mean that you physically can control. So you can physically control this and you can test that. The independent variable is the cause factor, meaning that it is the factor that causes the change, or it's the factor that will give you results. So it will lead to the dependent variable changing. So changing the light will cause the plant to grow. Removing the light will cause the plant to not grow or for the height to remain the same. So that's the manipulation of the independent variable. The independent variable is a variable that we can change or control in a scientific experiment, and that's the word I used in terms of change or control, referring to manipulation. It will represent the cause or reason for the outcome. So often in an investigation, we need to establish what is going to be the reason why that plant is going to increase in height, or what are the factors that make the plant leaves green. And so that would be the independent variable. So we've got to look at, is chlorophyll the factor that makes the plant green, or is it the oxygen and carbon dioxide that is used and produced by the plant? So we've got to establish exactly what is that factor that changes or causes the plant to actually become green or grow. And that's the factor that we refer to as the independent factor. Okay. The next variable is the dependent variable. And the word dependent means something that you can count on. Okay? So the dependent variable is one in which we can test the scientific experiment in order to get its values. So essentially, it's what's going to produce the results. The dependent variable is obviously dependent on the independent variable. So essentially, whatever changes because of the independent variable is dependent on that variable. So we refer to those as the dependents. So when a plant increases, gets more light, it's going to increase in height. So when it increases in height, it's because of the light. And so that height increase is dependent on the light, correct? It's the independent variable, which is the light, and the increase in the height is the dependent variable. Okay, so the factor that is being measured after the input variable has been enacted. Example, the temperature, the patient at different time intervals in degrees Celsius. So we talk about measuring the temperature of an individual, and you know that as the temperature increases, you feel much hotter. So the temperature of an individual increasing would be the dependent variable. So if someone had an infection and if you wanted to measure their body temperature because of that infection, measuring the dependent variable would be, let's monitor the body temperature over a period of time, and that would be because of the infection causing the change in the body temperature. So that's one way of us measuring the dependent variable. Okay, we're going to try and wrap this slide up by looking at the control variables. And the control variables are the ones which the scientists keep constant or hold constant during an experiment. Thus, we also know that the control variables are the constant variables, or sometimes referred to as the control only. However, control variables is not part of an experiment, but it doesn't affect the variables, but it is important because it has an effect on the results. Remember that it is not the same thing as the control group. So let's try and understand that in a nutshell. The control variables are things that you control and make the experiment fair. So if you are comparing the effect of light of two plants, you've got to ensure that you keep the amount of water the same, you keep the type of plant the same, you also ensure that the amount of soil that they kept in is the same. So those variables that you keep constant and you subject both the samples to are the control variables. And so that ensures that your experiment produces a fair test. And so as we end on the note of a fair test, I think you guys deserve a break, and then we'll look at what is validity, fair test, and trying to unpack some of the applications of these concepts. So a quick break, and then we'll see you guys at the end of that.